The overall goal of this procedure is to apply the Cook's kinetic method to determine the gas phase acidity of a cysteine polyalanine peptide. This is accomplished by first generating proton-bound cluster ions. The second step is to perform the collision-induced dissociation bracketing experiment. Next, the selected reaction monitoring experiment is performed. The final step is to analyze the data collected from the selected reaction monitoring experiments. Ultimately, the kinetic plot is used to generate the value of the gas phase acidity.
The main advantage of this technique over conventional methods like titration experiments conducted in a solution is that solvent effects can be eliminated.
This method can help answer key questions in the field of bioorganic chemistry, such as how acidic is a peptide in a solvent-free environment.
The implication of this technique extends towards diagnosis of effective acid-base properties of amino acid residues located in the interior of proteins, because solvent effects are eliminated.
Generally, individuals new to this method will struggle because the optimal mass spectrometry conditions are difficult to master without visual demonstration.
Though this method can provide insight into the intrinsic acid-base properties of peptides, it can very well be applied to other systems, such as peptide-mimicking polymers and oligonucleotides.
We first had the idea for this method when we wanted to study conformational effects on the acid-base properties of helical peptides.
Begin this protocol with preparation of sample solutions for the cysteine polyalanine peptide and 6 halogenated